So, as you all know, we have gathered here for the live webinar on sterilization of pharmaceutical product, the principles, practice, and validation. So, this webinar has been organized by Biomol.in. Now, Biomol.in is the online portal for dedicated for laboratory supplies. So anything that you need in a lab, starting from chemicals, microbiology products, cell culture products, or lab consumables, equipments, everything you can get it online on biomol.in. It just takes 3 steps: search, compare, and buy anything that you need for the lab. So moving on to the speaker, today we have Dr. Easwaran. He is the academic director at Biocon Academy. He has an extensive experience of 24 years in academia as well as industry. Previously, he has worked with some of the top companies like Merck, Biozine, and Biomedu in the field of biopharmaceutical production. And today, at the Biocon Academy, he has helped like 400 plus bioscience candidates to find the job of their dreams. So moving on to Dr. Easwaran, please let us know something about your work and then please start with the presentation.
Sure. Thank you, Dheeraj. For me also, it's really a good opportunity to know about biomol. And this is a new concept. I could see that there is a mall for the biological products and the supplies. So it's really good to know. And
Thank
I
you.
started my career as a lecturer in industrial microbiology, then moved into industry in production lines. And after 6 years of production experience, I moved into process development with Merck, and then into R&D and regulatory sciences with Biozine, and now working as a director for Biocon Academy. That's the big journey. And as a part of the career, the technical career, I picked up a lot of information in terms of what's unique about a biopharmaceutical product. Because initially there is a confusion in terms of understanding what is a biologic product and what is a biopharmaceutical product. And sometimes there is a overlapping term that is used as all are pharmaceutical products. So let me clarify a few things here before we start on the discussion, because that is required for understanding why specialized methods are needed for sterilization of biopharmaceutical products. First of all, the pharmaceutical products in general are the products that alleviate the illness or support the health. So, in general, you can call all the small molecules, large molecules, all put together as pharmaceutical products. There is no harm in that. But when it comes to the principles, practices, and how they work, it is completely different when compared to a small molecule to a large molecule. When I say small molecule, I am talking about the chemical pharmaceutical products. For example, let's say paracetamol, diclofenac sodium, or some antibiotics and something like that. These comes under the small molecules. And when I say biopharmaceutical products and biologic products, I'm talking about the large molecules. When I say large molecules, we are talking about molecules like insulin, erythropoietin, or GCSF, or interferons, or monoclonal antibodies, and so on. So what is the basic difference between this small molecule and the large molecule is that the small molecules are most of the times the chemically derived or chemically synthesized molecule, and their boiling point is very high. So even if you are going to use a sterilization method like autoclave, I don't know how many of the audience are familiar with the steam as a sterilizing agent. You can use steam as a sterilizing agent without any problem when it comes to the pharmaceutical product, which has a high boiling point, which means that the entire formulation do not have any heat labile compound. Whereas if you see the biopharmaceutical products, for example, if you see in this slide, I am taking examples of erythropoietin, insulin, and interferon alpha. So all these are having some common things like they have the beta-pillated sheets and the alpha helix. So these beta plated sheets and the alpha helix are the core molecule, or are belonging to the structure of the core molecule, which is the API, that is the active pharmaceutical ingredient. Proteins. Since they are proteins, the boiling point is very less. Because the boiling point is very less, the sterilization method has to be chosen in such a way that the sterilization method should not destroy this protein. As a part of sterilization, it should not destroy the protein. Now, quite often we are using this term sterilization. The term sterilization refers to the complete elimination of living organisms from the medicinal product, from our context. So when you say complete elimination, there are a couple of ways you can do that. Number one, you can put the whole thing into an autoclave, and the autoclave is used where it uh, allows the system to get 121 degrees centigrade at 15 pounds pressure. And in this condition, if you expose the product for about 20 to 30 minutes, then you can make sure that all the microorganisms are completely killed. Whereas when it comes to biopharmaceutical product, when you do that, this cannot work that way because these products are heat labile. The very API, very active pharmaceutical ingredient, like the erythropoietin, insulin, or interferon alpha, since they are proteinaceous in nature, they are extremely heat labile, which means that they are heat sensitive. Because they are heat sensitive, we cannot use the heat or steam as a sterilization agent. So if we cannot use steam as a sterilizing agent, then we have to look into what are the other options we have. And when it comes to pharmaceutical or biopharmaceutical product, generally you cannot take your own decisions, which means always you have to refer to a regulatory document which will give you some information about how to go about. For example, if I take this chart is from, if you can, I don't know if you can see this, it is EMA, that is European Medicinal Agency. They are giving this chart. This chart is called as a sterilization decision tree. So first question you have to answer is, if I have a product, for example, let's say I have a product called as insulin. First, I have to answer this question: can insulin be sterilized by moist heat at 121 degrees centigrade? So the question is, if you can use heat as a sterilizing agent. The answer is no. Why the answer is no is because if you use 121 degrees centigrade, the insulin will be denatured because it is a heat labile compound. So you are going to say no to it. If it is no, then can the product be sterilized by moist heat for 8 minutes, for 8 minutes at a lesser or little more of a time, more of a temperature, with a sterilization assurance level of 10 raised to minus 6? Again, this is not a good idea because normally for the biopharmaceutical products, we are looking at a sterility assurance level of 10 raised to minus 7 to 10 raised to minus 12. So if you are looking at that type of sterility assurance level, certainly exposing the product at slightly on a higher temperature at lower time is not a good idea because anyway it is heat labile. Now, can the formulation be filtered through a microbial retentive filter? This seems to be a good idea because this does not involve any heat. Since it is not going to involve any heat, but we have to see whether your formulation is amicable, whether the formulation is easy to be filtered through a microbial retentive filter. Because not all products can be filtered through a filter paper because of the viscosity. If it is water-like, then you can imagine it will be very easy for you to filter it through a filter paper. But when you consider a formulation, which means formulation means API plus excipient. API is the active pharmaceutical ingredient like insulin, and excipient is the non-drug component of the formulation. For example, it can be preservative, it can be pH stabilizing agent, or tonitizing agent, and so on, or bulking agent, and so on. So all these other components which are not the drug comes under the excipient category. So the active pharmaceutical ingredient plus the excipient forms the formulation. Now, when your formulation contains protein, there is a high probability that it is viscous. As you can imagine, the surface tension will be very high. Since the surface tension is going to be very high, then you have to see whether it is filterable, where your formulation is filterable. So if the formulation is filterable, then you can go for the use of a combined aseptic filtration and aseptic processing. Or if your filter, if it is not filterable, then you have to use a pre-sterilized individual component and aseptic processing and filling, which means that you have to fill the entire compound or entire formulation into a vial, and finally you have to sterilize it using a method which is not involving heat, which means it can be a gas sterilization like ethylene oxide, or it can be a gamma sterilization like using a gamma irradiation. If your product is filterable, then the method of choice is the combination of aseptic filtration and aseptic processing. When it comes to aseptic processing, the core of aseptic processing is the aseptic filtration, which means that after your entire production is over, in the next slide, I will show you how the production line looks like. When you actually see the production line, the product goes through a different set of methodologies to become pure. After the product become pure and it is formulated, there is a high possibility that you have added some microorganisms into it. So when you are actually using the filtration as a method, you have to have a good aseptic practice in place. So if you have a good aseptic practice in place, then the number of microorganisms which will hit the final filtration will be very less. If it is very less, then it is good for the process. So that is how we a sterilization method for the aseptic processing, or sterilization method for a given product. Okay, now I'm moving on into, give me 1 second. Okay, now I'm moving on into the biopharmaceutical process. So when you see a quick overview, or this is called as a block diagram, not even a process flow diagram. In a block diagram level, the whole thing starts with the inoculum preparation. After the inoculum is prepared, then it is transferred to a seed fermentation, so that this seed fermentation is capable of giving certain amount of required inoculum for the commercial scale production using fermentation as a platform. So for example, let's say I have an inoculum of E. coli, or I'll keep the insulin as a common example. Then if I have Pichia pastoris as an inoculum, then directly I cannot inoculate a small amount of Pichia pastoris to a production fermentation of 20,000 liters or 30,000 liters. So what you have to do is you have to have an inoculum, then you have to scale up the inoculum to a seed fermenter, so that the seed fermenter will create the necessary amount of cells that can be used as an inoculum for production fermentation. So once the production fermentation is completed, the entire amount of cell is harvested. Normally, if it is a microbial fermentation, the products are intracellular in nature. Since it is an intracellular in nature, it has to go through a cell disruption step here after harvest, and then directly it enters into ultrafiltration. So once it is going through the ultrafiltration, then it comes with the capture chromatography. The first chromatography is called as a capture chromatography, so that the maximum amount of protein, for example, in this case, if I'm taking an example of insulin, it can be a chain or a B chain. So the maximum amount of product will be captured in the first step, that is the capture chromatography. After capture chromatography, then you have to move the entire set of product to ultrafiltration, where you have to remove the buffer and change the buffer in order to the product to go through the secondary chromatography or a purification method. The secondary purification method can be an ion exchange chromatography, and because of that, you are actually changing the buffer. It is a buffer exchange step. where you are using ultrafiltration or tangential flow filtration method to change the buffer and concentrate. And the concentrated product is moved into the second chromatography, that is ion exchange chromatography, for example. Then you will have one virus filtration method. Virus filtration is not only a mandate in the cell culture, and nowadays it is a mandate for the fermentation also, because of the evident infection of the E. coli or some of the microorganisms with the heterogeneous or adventitious viruses. Then it goes through the final polishing chromatography, like hydrophobic interaction chromatography and so on. And then it comes to the ultrafiltration, the final filtration, and then it goes into the formulation. So it can be a ultrafiltration or it can be a sterilizing grade filtration. We'll come to this point. So once it is done, the final filtration is the membrane filtration. This step is what we are going to focus today on. How actually to do a sterile filtration, or how to sterilize a biopharmaceutical product using sterile filtration as a methodology before it is filled into the vial. As you can see here, it cannot be directly challenged to the final filter. The product cannot be directly challenged or cannot be directly filtered through a final filter, because it is a fouling product, which means the protein, it is a proteinaceous product. It has a lot of organic compounds attached to that. So as soon as the uh, fermented broth comes out, it goes through a depth filtration. This depth filtration is a clarifying filtration where most of the particles are removed. Most of the dirt is removed. Once the dirt particles are removed, when I say dirt particles, the large once that is removed, that filtrate is given to the depth filter or a surface filter. Surface filter is a pre-filter where most of the particles are removed, but not the microorganisms. Sometimes the particles along with the microorganism will be removed, but the intention of the pre-filter is not to remove the microorganism, it is to remove the particles. Once the particles are removed, we have only microorganisms, and that too in a very small level. So that is then given to the final filter. The final filter is otherwise called as a membrane filter or a sterilizing grade filter. This is responsible for sterilization of the biopharmaceutical product. Now we'll get to the details of mechanism, how it works. There is a distinct way in which the pre-filter works and the membrane filter works. The pre-filter works on a principle of adsorption. Adsorption means physical adsorption of the particles in the matrix of the filter, which means the particles are able to go into the filter pore, but it cannot come out of the filter pore. The reason is, if it is going through a filter pore, the filter pore is a tortuous path. Because it is a tortuous path, most of the particles are adhered or it is adsorbed within the matrix of the filter itself. Then you call it as a adsorption. So this is how the depth filters and some of the surface filters work. Whereas when it comes to the membrane filters, some pre-filters and some membrane filters work specifically in the mechanism of size exclusion. When I say size exclusion, what actually happens is that the size of the microorganism or the organic compounds is higher, or it is more than the size of the pore. Because of the size difference, all these particles or the microorganisms are eliminated in the surface itself. Because it is eliminated in the surface itself, it is called as a size exclusion method or size exclusion mechanism. So through which the microorganisms will be removed from the product. So only the product will be coming out as a filtrate. In that case, it will be a actually the depth filter is supporting the surface filter and the final sterilizing grade filter. As you can see here, the particles trapped in the filter depth, which means it is in the matrix. You can see the matrix of the filter, and the particles are completely adhered into the matrix of the filter itself, in the depth of the filter itself. That is why it is called as a depth filter. Here you are using a filtration media. The filtration media is a depth media, and mostly, as I was telling you earlier, it is large suspended particles is what is removed. Most of the time, the depth filters will have a larger particle size and also the pore size. Pore size is not small. Normally, the pore size will be very high, like 10 microns, 20 microns, 50 micron, 100 microns. When it comes to a pre-filter or a sterilizing grade filter, where there is a surface mechanism. When I say surface mechanism, as you can see here, all the particles are removed on the surface of the filter and no particle, which means no microorganism or organic particle is passing through the membrane. Only the solute is passing through the membrane. So in this case, that is how the membrane filter works. So most of the time, it is not a depth, it is a thin filter media that is used. Generally, polyvinylidene difluoride (PVDF) membrane is used in case of a membrane filter, and the pore size is very small and it is very narrow in nature. And as I was telling you, the surface filtration mechanism is used in pre-filtration as well as in the final filtration. So that is how this filtration mechanism works. As you can see here, I don't know if you are able to see the video. Give me 1 second, I'll see if I can play the video. I don't think. Okay, I don't think I'm able to play the video, but hello, if somebody is online?
Yeah, you'll have to upload that video separately. You can
I have
play it.
to upload the video separately.
Yeah.
Okay, but I'm afraid I have to search for the video. No problem, I'll explain this way only. Yeah. So as you can see here, the size of the organic compound and the size of the size of the bacteria is sometimes less, but along with the organic compound, and sometimes the size of the bacteria itself is high, then it is excluded in the surface of the membrane because of the size itself. Okay, so that is how the membrane filtration work. So through membrane filtration, the otherwise called as a sterilizing grade filter, you will able to completely eliminate the microorganism from the product stream. Then you call it as the sterilization step. So once the product is sterilized, then the product is ready for aseptic filling into the vials. Now, how to make sure that this sterile filtration is working all the time? So we have to consider certain things to validate. Validation is nothing but making sure. So how to validate the sterile filtration is through, as I was telling you, everything has to have a clear regulatory support for whatever you are doing in the industry. 21 CFR. CFR is Code of Federal Registry, and 21 CFR Part 11 113/B. This is giving a guideline what are the things to be considered to validate the sterile filtration. Number one, you have to find out whether the filter paper material, that is the material of construction of the filter paper, is compatible to the filter solution. For example, the compound, the formulation, the drug product, whether the drug product is compatible with the filter paper material. Because if the product is not compatible, then what will happen is a couple of things can happen. A, the product can damage the filter paper. If the product is going to damage the filter paper, then it is not sterilization because the pore size will increase, the product will just pass through. The other problem is it will clog. If the product is not compatible with the filter paper material, then the entire filter paper will clog. If the filter paper clogs, then there is no filtration happening. So either ways, you will not able to get the sterile filtration done. So the first and foremost thing you have to make sure is there is a compatibility between the product and the filter paper material. Second, before filtration, you have to make sure the filter paper pore size is as per the claim. For example, the filter manufacturer is claiming that it is 0.2 micron filter. If they are claiming that it is a 0.2 micron filter, how to make sure that it is still remaining as 0.2 micron before you start the filtration and after you complete the filtration? Both times you have to make sure that the membrane was having 0.2 micron pore size. Because if you are not making sure before and after, then you are not sure whether the product is actually sterilized or not. So there is a specific method for making sure that the filter paper is having a pore size as per the claim. This is called as integrity testing. In integrity testing, there are various methods like forward flow method or a bubble point method. If it is an aqueous product and the filter paper is hydrophilic in nature, in that case, what you can do is you can use a forward flow data or forward flow specification or bubble point specification, which is product specific, that can be generated in coordination with the filter manufacturer. Then you can figure out whether you are able to achieve a product specific bubble point or not. Once you achieve a product specific bubble point, then you can make sure, based on the value, based on the specification, if you are able to achieve the specification, then you are sure the pore size was zero point 0.2 if it is claimed as zero point 0.2 before and after the filtration. Then you have some assurance that the filter paper has actually worked well. Now, it is not only important or it is not only required correlated to the integrity value, that is the bubble point value, but also to prove that there is a correlation between the integrity value and the bacterial retention. If I take an example, if the bubble point value of one particular membrane is 50 psi, if it is 50 psi, which means if you achieve 50 psi and above, then you are sure that the membrane is 0 0.2 micron. This has to be correlated with the bacterial retention, which means in your laboratory, in the laboratory of the filter manufacturer, what they will do is that they will take a membrane, first find out whether the membrane is able to achieve 50 psi as a bubble point, and then challenge that filter with 10 raised to 7 CFU of Brevendimonas diminuta bacteria per centimeter square of effective filtration area, and to find out whether it is able to produce a sterile filtrate or not. If it is able to produce a sterile filtrate, then you can be rest assured that if in your production area, if you are getting 50 psi and above, then it will be correlated to 7 log reduction of bacteria because of the bacterial challenge test. So that is how the correlation is established between the integrity testing and the bacterial retention to prove that the sterile filtration has actually happened. The other important component or other important component which you have to consider is adsorption and binding. These two are correlated to each other in a way, because when you are seeing the compatibility of this membrane, sometimes the membrane will be compatible to the API, that is active pharmaceutical ingredient, but sometimes it so happens that the preservative, for example, let's say benzalkonium chloride, BKC, I'm just taking an example. The BKC has a very strong absorbing or binding property to any of the polymer. In that case, what happens is that the membrane will be compatible, but the BKC, that is the preservative in the product, will be completely bind or bound to the membrane. In that case, what will happen is your filtrate will not have microorganism, but it will have a very less concentration of the preservative, which is not acceptable. So you should not only prove the compatibility with respect to the overall formulation, but also specifically you have to prove that the components of the formulation is not absorbed or it is not captured by the filter material or the filter paper. In addition to all this, there is another important component, that is extractable and leachable. As I was showing you the process diagram, you can imagine that the product is going through a lot of harsh conditions, both in the upstream as well as in the downstream. In these harsh conditions, when the product meets some of the polymeric material, it extracts some chemicals, and these chemicals go into the product stream, both in the upstream as well as particularly in the downstream. So when it is going into the downstream, it is called as a extractable. And when this extractable mixes with the API or the excipient, then it's called as a leachable. Both extractable and leachable is a dangerous thing to have. But as far as regulations are concerned, what they have proved is that if extractable and leachable component, these are the chemical impurities. These impurities, if it is present less than a given value, then it is safe enough to be used. That means the product is safe enough to be used if the extractable and leachable concentration is less than the specification. So this is correlated to the clinical trial. In the clinical trial, they would have found out that in this concentration, it is not creating any critical side effect. It would have created certain known side effect, but it wouldn't have created any severe side effects. That is the reason why they have given some specifications. So if the product is meeting the specification which is less than the regulatory specification, then you can still go ahead with the extractable and leachable value. So these are all the components one has to consider to make sure that the sterile filtration is working very well for as a sterilizing method for a biopharmaceutical product. That is exactly what I wanted to share with you as far as the technical area is concerned. And I'll take another couple of minutes to let you know what we are doing at Biocon Academy. What we are doing at Biocon Academy is that we are training the students who have completed their degree courses in engineering, which is particularly BTech in Biotech or MBE in Biotech, or it can be MSc in Life Sciences. It can be MSc Biotechnology or MSc Microbiology or Biochemistry and so on. We take the applications from these students and then we interview them in two ways, that is HR interview and a technical interview. If they are passing these interviews, then we admit them into a short-term course. The next slide, I'll show you what are the courses we have, and we admit them into a short-term course, different courses. So we are collaborating with education partners like Keck Graduate Institute in California or BITS Pilani in India, and one more education partner I'll show you in the next slide. On one side, we are collaborating with the education partner to give the theoretical background of the subject. For example, it can be biotechnology, or it can be clinical development, or it can be microbiology, applied industrial microbiology, and so on. This happens in the morning. In the afternoon, the participants will go into Biocon and work for 2 weeks in each of the laboratory. It can be quality control, quality assurance, production, R&D, regulatory affairs, and so on. So in each department, they'll work for 2 weeks. So they have a theoretical background created, and now they are able to experience and how the theory is applied onto the lab. And we have a component called as hands-on training that happens in either Thermo Fisher or at Biozine, which provides a or corresponding to the programs what we are talking about. It is not only important to create a strong technical background through these three components, it is very important to create a strong professional skills, because communication skills is very important, because sometimes the students know the science, but they are not able to articulate their thoughts into the words during the interview, and they do not get a meaningful job because of that. So it is very important that we have to create a strong professional skill. So the professional skills development training is a part of the overall Biocon Academy training. Now, after completion of this training, we conduct the placement drive. So in the placement drive, you will see Biocon, Syngene, and other companies participate. When I say other companies, more than 40 companies are 40 core companies are participating, and they are hiring the students. I'll show you the next slide with the complete data. The first program what we have launched is in the area of biotechnology in the name of biosciences. So in this, we have completed 13 batches and 14th batch is in progress. All 13 batches has been 100% placed. When I say 100% placed, we did not have even one situation where we have to move one student from batch 1 to batch 2 or any cross batches to place the student. Every time by the end of the 4 months training, immediately they get placed in the industry. So with this success of biosciences program, then we launched the industrial microbiology program, as I told you, applied industrial microbiology program, and this is in collaboration with BITS Pilani. Biosciences is in collaboration with the Keck Graduate Institute in California. The next batch, that is batch number 15, is starting on 18th of July 2019. And the microbiology program is starting on 26th April, for which we are doing, for both of it, we are doing the admissions now. Then we did one bioscience management program for the Biocon employees. And then we launched a detailed program, a 2 weeks detailed program for the faculty, that is the lecturers and professors who are teaching biotechnology in the colleges and the universities. So we have completed 2 batches of faculty development program with the same model, where in the morning they will have theory sessions and in the afternoon they will go into the Biocon to see what actually happens in the industry. So they will able to teach, or their orientation of teaching in the application side actually increases. And we are doing admissions for that, and the program is starting on 1st July 2019. We have also completed 1 batch on clinical development program, which is specifically targeting the pharmacy students. So biosciences is for biotechnology, microbiology students. The applied industrial microbiology is for, again, biotechnology, microbiology, life science students. The clinical faculty development program is for the faculty of biotechnology. Clinical development program is for the pharmacy students. And that is going to start on 1st July, that is faculty development program. Clinical development program is going to start on 30th of August. Now we are going to launch a new program, that is Biocon MS Ramaya Certificate Program in Quality Control Analytical. This Quality Control Analytical is going to revolve around the QCA, that is Quality Control Analytical Department, the skill sets that are required for quality control. Most of the aspects of instrumentation, regulatory requirement, and practices are going to be covered in this program, which are useful in the quality control laboratory. This program we are going to launch on 16th of August 2019. So these are all the programs what we have right now, and these are the dates for the next batch, and this is the new program what we are launching at Biocon Academy. Thank you very much for your patient listening. And if you want to know about Biocon Academy, you can visit our website, that is bioconacademy.com. If you have a question, you can email your question to admissions@bioconacademy.com. Or if you want to talk to us, you can pick up the phone and dial 888-4899-898, and you will get the answers from us. So thank you very much for your patient listening, and I open the session for the questions. You can type
Thank
the
you very
questions in your
much,
chat box.
Thank
Sorry.
you very much, Easwaran. It was a very nice presentation. And
Thank
I
you.
myself have one question actually. So, do
Please
you
go.
have any course at Biocon which is meant for people who are already in the industry professionals?
Yes,
Uh,
there are a couple of things we are doing. One is these programs, whatever programs I have shown you in the last slide, all these programs are attended by both industry professionals as well as the students. Only difference is that if you are in the industry, or if you are actually working at present, then you cannot attend the program. You have to be away from the industry. If you are, for example, you have taken a break and you want to go back to the industry, then you can attend these programs. So it is not for the ongoing industry professionals. It is specifically to help the students to gain the industry entry in
okay.
first place. Yeah.
And mainly it is for graduates or postgraduates, any kind of eligibility.
Yeah, it is program specific. As I was telling you, for the biosciences program, it is BE Biotechnology, BTech Biotechnology, MSc Biotechnology, MSc Microbiology, Biochemistry.
Okay.
These are the eligibility criteria, and B Pharmacy. And for microbiology, it is basically MSc Biotechnology, Microbiology, or B Pharmacy. For clinical development, only B Pharmacy. And for faculty development program, you have to be teaching in the university or college.
Right. So,
Any other question?
yeah. If you have any question, please type it in the chat box. Yeah, Rekha, please go ahead with the question. Oh, Hemant, okay, there are two questions.
Okay, 1 minute. Rekha had a question regarding the earlier session. Okay. How are viruses removed from the product? Excellent question. Rekha, as I was telling you, if you remember the process flow, I mean the block flow diagram, what you have used, there are a couple of virus removal steps. The virus can be removed using three technology. One, acidification, that is low pH removal. Second is chromatography, and third is nanofiltration. You have to use all these three technologies in every process. So there is a regulatory requirement by CBER, that is Center for Biologics Evaluation and Research, as a part of FDA. They have recommended that all three or orthogonal steps has to be used to remove viruses. So you have to use acidification, you have to use chromatography, and you have to use a nanofiltration step to remove viruses from the product. Does it answer your question, Rekha? No problem. Any other questions? Hemant, sugar and protein sterilization technique. Hemant, the complete difference between a sugar and a protein is based on their biochemical nature. Sugar is a carbohydrate. Because it is a carbohydrate, the method which is used for sterilization of sugar as a solution can be different. You can choose to use the filtration as a method for sugar solution. Absolutely, there is no problem. As long as you can prove that the sugar is not uh, binded, it is not bound by the filter material itself. Because sugar has a capability to bind to the material. If it is binding too much to the material, then you have to use a specific type of filter to completely filter or to sterilize the sugar. As far as protein is concerned, the most preferable method is membrane filtration method itself. Does that answer your question, Hemant? No problem. Yeah, Neha. Oh, thank you very much. Yeah, please go on. When is gamma irradiation or chemical-based sterilization is used in biopharmaceutical products? Okay, very good question. If you would have, uh, if you would have imagined or if you would have internalized the validation considerations, it would have got answered there. Neha, if you remember, we said that there are products which will bind to the filter material. If the product will bind to the filter material, you cannot use, even though the product is compatible, even though the product is compatible to the filter material, but because of the binding property, you will not able to use the membrane filtration as a method. In those cases, the gamma irradiation is used or chemical sterilization is used for a biopharmaceutical product, which means the product is heat labile. It can be filter sterilized, but the filter compatibility is there, but not the filter binding studies is cooperating. In such cases, we can use the gamma irradiation as a specific method. If you want to take some examples, you can take some examples like uh, biopharmaceuticals, which has nuclear decay components. So these nuclear decay components with biopharmaceutical products, which will always have the problem of binding to the membrane. So you will lose these components. So generally, these components are used sterilized using gamma irradiation as a method. Does it answer your question, Neha? And in the meantime, I have a question from Heyman. What about sugar plus protein formulation? So if you have a sugar plus protein formulation, then it is very easy to handle, because when it is a sugar and protein, the concentration or the viscosity is very high. If the viscosity is very high, it is not a good idea to use the membrane filtration as a method. And if it is a combination of sugar and formulation, as I was telling Neha, this is another good example where you can use other than filter sterilization as a method for sterilization of the product. It can be gamma irradiation or it can be chemical sterilization like ETO. The question here is generally when it comes to biopharmaceutical products, the combination of sugar and a protein is very less. Products. Does it answer your question, Hemant? No problem. Thank you for good questions. Any other questions from anybody else?
Yeah, so uh, when you're using gamma irradiation, would it affect the chemical structure of the formulation in any way?
That's also a very thoughtful question. When you are choosing the gamma irradiation, you have to choose the strength of the irradiation. If you are using, for example, 25 kilograms of gamma irradiation, it will not affect the chemical stoichiometry of the molecule. But if you are using 50 kilograms, thinking that you are going to overkill the microorganisms in terms of validation, then you will have a problem of having a stoichiometry change in the protein structure because of the gamma irradiation. So it is more to do with the selection and validation of the specific strength of gamma irradiation for sterilization of the given product. Does it answer your question?
Yeah, yeah. Thank you.
Yeah, no problem.
So, do we have any more questions?
Any questions, Kausalya? Any questions, Pratiksha, Sachin, Tejasvi, Amit? Any questions?
Hemant is typing.
Hemantis? Oh, okay. Thank you. Good. I think we had a good 1 hour discussion.
Yeah. I think one more message is coming up. Let's see.
Okay, okay. I'll wait. Pratiksha is done. So by the time somebody types, Thank you, Viraj, really, for creating this opportunity to discuss with a lot of people. Thank you. Just want to know your views about sugar solution sterilization at less time. Ah, this sugar solution seems to be a problem. So this sugar solution, if you are using as a solvent, um, Pratiksha, if you do specific gamma, does it affect the nutrient or material of medium? Normally at 10 minutes. The time is not a problem, Pratiksha or Hemant. What actually happens is when you are using a gamma radiation, we have to validate both, or rather, three things you have to validate. One, you have to validate what is the minimum strength that is required for removing microorganism at a given exposure time. So all these three things are together. It cannot be considered separately. So if you think 25 kilograms for 10 minutes will remove 10 raised to 6 CFU of microorganisms, then you have to use a bioburden approach to prove that what is the given strength will remove how much of microorganism. So at the time of validation, what you have to do is you have to spike the product. For example, let's say sugar. You have to spike the sugar solution with a known concentration of bacteria, CFU, and then put it under the gamma irradiation or put it through a gamma irradiation. And you have to validate different timeline. You can start with 5 minutes and 7 minutes and 10 minutes and 12 minutes and 15 minutes and figure out what is the concentration or what is the dosage of gamma irradiation required for removing 10 raised to 6 or 7 CFU of microorganism at a given, or what is the minimum amount of time that is required. So you have to validate all the three together using the validation experiments. Uh, you are right. Absolutely, you are right. Your understanding is correct, Hemant. You cannot do gamma irradiation in the laboratory stage. You have to find the right set of people who have been authorized to do studies on gamma irradiation as a sterilization method. Because using gamma radiation requires a lot of regulatory requirements and safety requirements to be used as a trial as well as a method for sterilization. So, as you rightly mentioned in your message, not every laboratory can use a gamma radiation to take a trial. That's right.
So then I think he also means like if he wants to do sterilization in the lab, what should he go with instead of gamma
See,
irradiation?
no, it is something like this. If you are having very less choices, for example, let's say I want to sterilize a product, which is a sugar-based product or sugar-based formulation with protein, and if the requirement is sterilization, because quite often we get confused between sterilization and reduction of bioburden. Because every time when you mention that your product is a sugar-based product with a protein, we need to understand whether it is a sterile product. When you say sterile product, if it is purporting to be sterile, then certain ways in which it has to be taken. For example, let us take injectable. The injectable is purporting to be sterile. So are we talking about a injectable compound which is a sugar-based solution with protein? In that case, then the validation is completely different. Then you don't have a choice but to go to centers like BARC, that is Baba Atomic Research Center, to do the validation, because they are, and I don't know if you have some more laboratories or some more centers. But as of now, what we need to understand is whether the product is supposed to be sterile or we are talking about bioburden reduction. If the product is orally consumed product, then it is not a problem. Yeah, he is giving another option, that is BRIT in Hyderabad is doing the trials. So you have to go to such type of centers to do the trials. But we need to be very clear in the end objective. If the end objective is bioburden reduction, then probably gamma irradiation is not a requirement. You can do away with heat as a sterilizing agent. You can use sterilization or pasteurization to reduce the number of microorganisms in the product. Thank you, Hemant, for the input. BRIT Hyderabad. Can we do sugar sterilization in the lab at 121 at 10 degrees centigrade, 10 minutes? Okay, 121 degrees centigrade for 10 minutes. So it is little difficult because, as I told you, sugar is a carbohydrate, and more than 100 degree, normally it will char. The charring of the sugar is a big problem. When you actually put a white sugar, you come out with the brown color sugar. I don't want to use the term brown sugar, but I can tell you it's a brown color sugar because of charring. Uh, but this problem is little less when in the moist heat as a sterilizing agent when it compared to the uh, compared to the moist heat, compared to the dry heat. But if you want to sterilize sugar solution, then I would like you to consider the purpose of sterilizing the sugar solution. Are you using, or probably are you going to use the sugar solution as a solvent for making a drug product which is going to be an injectable? Only if the answer is yes, then it is worth doing all these trials. You're welcome. Any other questions? Bittu has just joined about 5 minutes back. Bittu, to give a quick drill of what we have done is we went through the method of sterilization of biopharmaceutical products. Particularly, we dwelled through the filtration as a method for sterilization of biopharmaceutical product because they are heat labile in nature. And also, we went quickly over what are the programs, what we do at Biocon Academy. And now we are doing the discussion part of it. But still, thank you very much for joining us.
We shall share a recording of the entire presentation so that you can
That
go
will be
through it again.
wonderful.
Yeah.
Is that all? Shall we close the session? There are no
Yeah,
questions.
I guess so. There are no more questions, so then we should close the session now. Thank you very much, Dr. Easwaran, for your
No problem.
wonderful presentation and expertise on the subject. So, as I said again, I'll be sharing the recording of the entire presentation, entire webinar, along with the question and answer session, so that you can always come back to it. And I believe sir would always be happy to answer the questions at a later time if anything comes up. Thank
Yes.
you so much.
You're welcome. Bye to you.
Bye.